Hey class, how's it going? So this is going to be a short and sweet little lecture. This is just the last few slides from our Tuesday lecture that we didn't get to go over. So on our Tuesday lecture, we learned about cells. The lecture before that, we learned about nutrients. So chapter 3 is nutrients and cells. And then the last part, which we're going to look at right now, is how do those nutrients get into the cells? So this is membrane transport. So just to remind you, substances must travel through the plasma membrane or the cell membrane in order to get from the outside of the cell into the cell. So this would be macronutrients like water, carbohydrates, amino acids, and lipids, which are all necessary for cell metabolism, which is just the chemical reactions in the cell. And vitamins and minerals, which also help with chemical reactions in the cell. But the problem is that the phospholipid bilayer is selectively permeable. Remember, that means that it is choosy. It only lets certain things pass through. So large substances like the green macromolecule in the figure and polar substances, which are things that have charges, cannot pass. Whereas nonpolar molecules and small molecules pass easily through that hydrophobic interior. So remember, the core of the lipid bilayer, the interior of it, is hydrophobic because it's made up of these nonpolar phospholipid tails in yellow in the figure. And so, I mean, imagine that's like bacon grease, right? That interior, and so it's oil. It's not oil, but it's like oil. So things that have charges won't be able to pass through because charged substances don't interact with hydrophobic nonpolar substances. So water is an exception to this because we know that water is polar, but it's small enough to where it can pass through all right. So there are two main mechanisms that the cell uses to transport molecules through that plasma membrane, through that hydrophobic core. There's passive transport, which requires a concentration gradient, which is basically, as it sounds, a gradient of concentration. So there's higher concentration of whatever molecule on one side compared to the other. So molecules tend to move from areas of high concentration to low concentration. So this requires no additional energy. Active transport requires additional energy in the form of ATP because if you look at the diagram there, that potassium ion, that K plus, is moving through the plasma membrane through a protein channel from an area of low concentration to an area of high concentration. So that's like going against the grain or against the flow. So if you're going to go against the flow of a current, you need to put energy into that. So active transport goes against the flow and requires ATP, additional energy in the form of ATP. So let's look at these two mechanisms. Again, passive transport does not require additional energy. The molecules move through the plasma membrane down their concentration gradient. So that it goes with the flow from an area of high concentration to an area of low concentration. And so you have three types of passive transport. You have diffusion, where literally these small nonpolar molecules will diffuse through the plasma membrane using that concentration gradient. You have facilitated diffusion, where molecules that are charged or that are larger that can't normally pass through the plasma membrane will pass through, facilitated by these protein channels here in green, the transport proteins. And these are still moving down their concentration gradient. And then osmosis, which is like diffusion, except specifically water. Osmosis is the diffusion of water cells. So we're going to look at these three forms of passive transport. Remember, this does not require additional energy other than a concentration gradient. So small nonpolar molecules move down their concentration gradient through the plasma membrane. Nonpolar molecules can just pass through that nonpolar core, that hydrophobic core, just fine. Facilitated diffusion uses it is the diffusion of polar and charged particles. So these particles are still moving down their concentration gradient from high concentration to low concentration, but because they're polar and charged and maybe a little bigger, they can't pass through the membrane, so they need the help or the aid of these protein channels, these transport proteins in green, that basically are like poking holes through the plasma membrane. And each transport protein is specific for a certain molecule, either whether it be an amino acid or calcium ions, which you can see there as little blue balls, or glucose molecules, which you can see there as red hexagons. And it's just important to note that this still uses a concentration gradient. This is still passive. There's no additional energy being given. You're just poking a hole through the membrane with this transport protein, and these molecules move down their concentration gradient. And so they do not require, this does not require ATP. And then osmosis is just diffusion, but of water. And again, using a concentration gradient. So in this picture, you have a cell. If there is a higher concentration of solutes outside the cell on the top, so basically if there are more ions outside the cell than there is inside, water is going to diffuse out of the cell to try and equilibrate the concentration of solute outside and inside. And so since water diffuses out, the cell is going to shrivel. On the bottom example, if there's a higher concentration of solutes inside the cell compared to the outside, water is going to diffuse into the cell to try and dilute or reduce the concentration of solute inside so that it can equilibrate it to the concentration of solute outside the cell. And when that happens, the cell can actually burst or lyse. Remember, we saw the root word lyse in hydrolysis, which is the chemical reaction that splits apart polymers. And lyse means to break apart. So because the water is diffusing into the cell via osmosis, eventually that cell is going to be like a balloon that just keeps being filled with water and then bursts eventually. All right, active transport uses protein channels to move molecules against their concentration gradients. So this is going against the grain. This is going against the flow. It goes from low concentration to high concentration. So in order to go against the flow, like if you're going to try and canoe upstream a river, you're going to need to put energy and work your muscles, right? So if you're going to try and move a molecule like this potassium ion against the flow, against the concentration gradient, you're going to need to give energy. And so this active transport is powered by energy from ATP. All right, and the last forms of transport that we'll look at two more. And these are exocytosis and endocytosis. So these happen when cells need to import or export large amounts of substances. All right, so exocytosis transports large amounts of substance outside of the cell. Easy to remember. Exo means outside, right? Exoskeleton means an outer skeleton. And this happens when a membrane-bound vesicle fuses with the membrane, with the plasma membrane, and then expels a large molecule or large amounts of molecules outside of the cell, as you can see in the figure there. So this happens during milk secretion, as we looked at in class, and neurotransmitter release in your brain. So when neurons fire and then they release neurotransmitters, those neurotransmitters are released using exocytosis. Endocytosis is the opposite, so it transports large particles into the cell. So this has more to do with size than it does with amounts. And the opposite happens and endocytosis. Remember that endo means inside. So this is bringing something inside the cell. So for this, a vesicle pinches the plasma membrane inward and brings the large molecule into the cell. And this is what happens when your white blood cells engulf a bacteria or eat a bacteria to get rid of it. It does this by endocytosis. All right, so let's watch a little movie that animates these different forms of cellular transport so that it can solidify the concepts that we just learned. Then we'll do a few questions and we'll finish chapter 3.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
All right, so hopefully that little video helped solidify the different concepts we learned. I love video, I love animation because it really brings to life these kind of static concepts. So I really do encourage you guys, go onto our Mastering Biology page and you can actually find animations and videos for all of the chapters that we are going to be in, and it'll just help a lot. You can also go to YouTube. There's a few YouTube channels that I really like for biology. So you can go to Crash Course videos and a few other videos. If you just Google some of these things, you'll pull up some pretty helpful animations. Khan Academy, that's Khan Academy. They also do little online lectures like we're doing on different biology concepts. Okay, so let's do a few questions and then we'll wrap up. So question 1, cholesterol is a molecule in animal cell membranes. Since cholesterol is hydrophobic, where is it most likely to occur? On the surface of the lipid bilayer or the interior of the lipid bilayer? So for this question, you have to know that the surface of the lipid bilayer, either surface, is composed of the hydrophilic head of the phospholipid. It loves water, so it's on the outside surface. This hydrophilic head is polar, meaning that it has a charge, in this case a negative charge from a phosphate group. And then the little tails there are hydrophobic. They're nonpolar. They have no charges, so they hate to interact with water, and they stick together on the inside of the lipid bilayer. So cholesterol is also hydrophobic. It's afraid of water because it's nonpolar. Anytime you see the word hydrophobic, that more or less means that the molecule is nonpolar. So this nonpolar molecule is not going to want to hang out with the polar heads in green. It's going to want to hang out with the other nonpolar tails in the center of the lipid bilayer. So the right answer is the interior. How many of these features does a typical bacterial cell have? So let's walk through each one of these. Do bacteria have DNA? Yes. All living things have DNA. All cells are going to carry DNA. That is a universal component of cells. Does bacteria have a cell wall? Now, not all cells have a cell wall, but bacterial cells do. And so do plant cells as well as fungi. So anything that you think of as plant-like, plants and fungi, and bacteria, they have cell walls. Do they have a nucleus? They do not. So bacteria are prokaryotic. Prokaryote means, pro means before, karyotic means nucleus. So prokaryotic cells, such as bacteria, are cells that are thought to have come about before cells had nuclei. Okay, do they have ribosomes? Yes, ribosomes are a universal component of cells because all cells need to make protein, and ribosomes make protein. Do they have a cell membrane? Yes. Again, cell membranes are a universal component of all cells, prokaryotic or eukaryotic. So bacteria then have DNA, cell walls, ribosomes, and cell membranes. So those are 4 features common to bacteria. Okay, so now talking about membrane transport, what process allows charged amino acids to enter the cell? So let's work through these. You can right away cross out B, osmosis, because osmosis is the diffusion of water molecules. And we're talking about amino acids, so you can cross off B. You can also cross out D, exocytosis, because exocytosis is talking about exporting something outside of the cell. So even if you had no idea what exocytosis is, you can know by the word exo that it's talking outside of the cell. So we're wanting to bring amino acids into the cell, so you can cross out D. And you're left with A and C. Now A, diffusion, is a passive transport, but only of small or non-polar molecules. Amino acids are large and polar. So they're going to have to enter the cell through the plasma membrane using facilitated diffusion. And that's when you have a protein channel or a transport protein, same thing, right? Those two things, which pokes a hole in the plasma membrane to allow these charged molecules to pass through. So C is the right answer. All right, last question. What best describes active transport? So here is a figure of active transport from the textbook. And let's just read through each one of these. The potassium ion will move from high concentration to low concentration, and ATP is used. That is wrong. As you can see from the figure, the potassium ion is moving from low concentration to high concentration. It's moving against the concentration gradient or against the flow. So A is wrong. B, the potassium ion will move from low concentration to high concentration. That's right. And ATP is used, and that's right as well. So whenever you move a molecule against the concentration gradient, much like going against the flow of a river, you need energy, in this case ATP. Let's look at C and D as well. C, the potassium ion will move from high concentration to low concentration. You know that's not right. And it says ATP is not used. You know that's not right either. In the diagram, it shows you that ATP is being used. And you should know that active transport is active. It needs energy, so you use ATP. And then D, potassium ion will move from low concentration to high concentration. That's right. ATP is not used. Wrong. So yeah, the right answer is B. All right, guys, hope you enjoyed this lecture. Finish chapter 3. Chapter 3 homework is already posted, so go ahead and have fun with it. Look over that test review. Email me a few questions that you might have trouble with so that we can go over that on an online test review that I will be posting tomorrow. All right, have a great rest of the week, and I guess I'll see you on the internet. And for sure, see you on Tuesday for our test, bright and early at 8 a.m. Take care.